So Learning Unit 9 is on enzyme kinetics. So we've kind of built to this point with Chapter 6, learning about protein structure, Chapter 7, to some extent, starting to learn about how enzymes can have function with hemoglobin and thinking about binding, and then enzyme catalysis in our last learning unit, and finally sort of now understanding, well, enzyme catalysis has rates of those enzymes. And so we think about the important kinetic parameters that we'd like to know for an enzyme. There's really two that we're interested in knowing. Vmax, which is how fast the enzyme can go, its maximum velocity, and then Km, which reflects the inherent affinity of the substrate for the enzyme. Mathematically, you can also think of it as being the substrate concentration you need to be at for the enzyme to be working at half of that maximum value. Now, the problem is both of those parameters, although we're interested in them, can't be measured in the lab. So like we often see, we measure things that we can measure, like substrate concentration and initial reaction velocity, and then we manipulate our data so that we can determine Km and Vmax. And that's all about what the Michaelis-Menten equation is. And so we spent some time understanding Michaelis-Menten kinetics, understanding how to analyze data using a Lineweaver-Burke analysis, again, a way to get data as a straight line, so we can get information like Vmax and Km from the slope. Second to this is we sort of moved into, well, now that we understand how enzyme kinetics work, let's now put in an inhibitor and ask how things happen and how things change when we have, for example, a competitive inhibitor or we have an uncompetitive inhibitor. So again, two different scenarios that we looked at, and we looked at examples, real life examples like alcohol dehydrogenase in terms of thinking about how these processes worked. The last thing we then went into in our chapter on enzyme kinetics is thinking about how we regulate enzyme activity. And so we talked about, we can regulate enzymes both with availability and activity. So availability is really a long-term sort of measure, controlling the amount of enzyme that's there or not there by regulating the rates of synthesis and degradation of enzymes. But that's not efficient in the short term. In the short term, it's actually more efficient to regulate the activity of an enzyme by turning it on or off. So we learned about mechanisms for how that happens with covalent modification and allosteric regulation. And then we actually mapped all of that together onto a real-world example looking at glycogen phosphorylase. So again, the enzyme responsible for breaking down our storage polymer of glucose as glycogen and breaking it down into glucose or glucose 1-phosphate so that we can then use that as an energy source. So three main things that we kind of dealt with in this chapter, a good bit more math that we've kind of dealt with in our previous chapters, but then kind of rounding out at the end, kind of pulling things together with enzyme catalysis and thinking about how we regulate an enzyme.